only have one. So if you just tried to make these little stair steps across the two sides of the double helix randomly, you'd have some stairs that are wider if you got, for example, two purines that were big molecules together, and you'd have some stairs that were relatively narrow if you got two pyrimidines, which are smaller. So the outside of this staircase, of this spiral, would be going in and out, which was not structurally stable. And that's where Chargaff's rule came in. Because they realized that the implication of Chargaff's rule, the fact that there's always the same amount of adenine and thymine and always the same amount of cytosine and guanine, suggested that it may be that one always pairs up with the other when they're matching together on the inside of the helix. And it turns out when they looked at how these kinds of nitrogenous bases would match up, they found that those particular combinations, A and T and C and G, would always maximize the potential weak bonding, a kind of hydrogen bonding that occurred between those nitrogenous bases. So they'd actually solved two problems. They had figured out how you can have a regular distance along the whole length of the staircase, and also in the treads, and also they figured out what could hold the staircase together, because if you always matched A with T and G with C, you would maximize the potential weak bonding that would hold the two sides together. It's interesting that there's a third property that comes out of that we'll talk about in a second. Now, in April 1953, which is only a year after people became convinced that DNA was the information molecule, Watson and Crick published a very short, a one-page paper in the British journal Nature, which is one of the more prestigious scientific journals in the world, which described the double helix. As I said, arguably one of the most important discoveries in the 20th century. They described the molecular structure, they described how they thought it would all fit together, how it fit the X-ray data, and so forth. But the real significance of this work was not simply to describe the 3D structure of DNA, but instead to show how that 3D structure, to suggest how that 3D structure might actually say something about replication. This paper is remarkable. It ends with the following sentence, which I love. It's sort of British understatement. The last sentence is, and I quote, "It has not escaped our attention that the specific pairing we have postulated," and what they're referring to that is the A with the T and the G with the C, "immediately suggests a possible copying mechanism for the genetic material." The fact is that the fact that A and T and G and C were always paired together meant that if you took the two sides of the molecule apart, you would always know what the other side have to be. We're going to talk about that in a second. That's called complementary base pairing. And it was this fact that suggested the mechanism by which DNA was replicated. Now, just later that year, in a subsequent longer paper, Watson and Crick elaborated on this copying mechanism. And what they showed was that, what they suggested was that it was this so-called complementary base pairing where A and T, and C and G were always matched together, that meant if you could somehow unzip the molecule, take it apart, and have the two halves separate, you could always retrieve the sequence in the complementary half that had been removed. You would always be able to say, if there's an A on this side, then the other side must be a T. There's a C here, the other side's got to be a G, and so forth. Now, they proposed then that what happened during replication is that the old DNA strand, when these two, an old DNA strand could serve as a template for two new DNA strands by having the old DNA strand separate and having the opposite sides built on to the existing strand, creating two new DNA strands. And thus, the information in DNA would always be duplicated exactly. And at the end, we'd be left with two double helices that were copies of the original single double helix. Now, I want to stop for a second, or pause for a second, and give you a little bit of a historical note. As you can imagine, this was a very important discovery, and it was awarded a Nobel Prize. The Nobel Prize for Physiology and Medicine in 1962 was awarded for the discovery of the double helix of DNA. Who got the prize? Watson and Crick, of course, because they had published the paper that described the structure, and also Maurice Wilkins, who was the biochemist who had purified DNA. Interestingly, Rosalind Franklin, the woman who actually made the crystallograph, which Watson had seen and which provided the essential information for understanding the structure, was not awarded the prize. This has been the source of some interest and controversy over the years. Why not? Much has been written suggesting that, in fact, Franklin was denied credit for what was probably this most important contribution of the double helix. Certainly, it was well known that her relationship with her colleague Maurice Wilkins was a little bit rocky. Wilkins assumed that when Franklin showed up to work in the lab that she would be more or less his assistant, and that wasn't her attitude. She had a very difficult social interaction with most of her co-workers, and she was eventually asked to leave the lab. And she was told at the time by the head of the lab that she should stop her work on DNA. Franklin did actually publish one more paper on DNA. It turns out it was a paper that helped confirm the Watson and Crick theory. But then she put down her work on DNA and she began a completely different career, using her X-ray crystallography skills to look at things like the structure of viruses and so forth. It's also true that if you look at accounts of the discovery of DNA, say in the 60s and the 70s, the contributions of Rosalind Franklin are relatively understated. But the fact that Franklin did not share the Nobel Prize actually has to do with something other altogether. And the fact was that she had died before it was awarded. Nobel Prizes aren't given posthumously. Franklin died of ovarian cancer in 1958 when she was only 37 years old, 5 years after the description of the double helix by Watson and Crick, and 4 years before the Nobel Prize was awarded to Watson, Crick, and Wilkins. Fortunately, in recent years, a number of biographies and other kinds of historical analyses have made it clear what the critical importance of Franklin's contribution to this work was, and she now figures prominently in just about any textbook or description of the process. Okay, well, let me put that aside, and I want to go on, and I want to go back now to this proposal that Watson and Crick had for how the double helix might be replicated because of this complementary base pairing. Watson and Crick suggested that the molecule must unzip, and then each half of the molecule could serve as a template for a newly formed half. It's a good hypothesis, but was it correct? Actually, as soon as Watson and Crick proposed that, there were two alternative hypotheses very quickly proposed. The first alternative suggested no, the DNA double helix must remain completely intact when it's replicated. That is, the two strands don't separate, and the entire molecule is used in some fashion as a template for making more DNA. This alternative was called the conservative hypothesis of replication, or the conservative model of replication, because the original DNA double helix molecule remained completely intact and conserved. The idea must be that there was some intermediate molecule that got information from the structure of the DNA helix and used it to build a completely different helix. Now, a second alternative suggested that the original DNA molecule becomes completely broken down during replication, with the newly copied DNA in the right order assembled by some unknown mechanism. In other words, the DNA double helix in this model would actually be irrelevant. It didn't tell us anything about how we might copy DNA, it just happened to be a double helix. This alternative was called the dispersive model. It was called dispersive, obviously, because the DNA in the original helix that's copied just becomes dispersed and incorporated wherever in the new copies that are created. Now, based on what was known about molecular biology of DNA in the 1950s, both of these hypotheses were reasonable as any other, although neither had as neat a solution to the problem of duplicating the exact order of the nucleotides in the strand of DNA, this order being the information that we're seeking. Now, in hindsight, knowing what we know about DNA today, both of these alternatives actually seem quite unlikely. But their value then, and still now, was to serve as alternative hypotheses against which to test specific predictions made by the Watson and Crick model. Because what we now want to do is to go beyond the hypothesis that Watson and Crick had about how DNA was replicated to testing specifically whether that hypothesis was true. The mechanism, by the way, that Watson and Crick proposed became known as the semi-conservative model of DNA replication. This was called semi-conservative because it predicts that during replication, the double helix unwinds, and now the new daughter helices will both have one strand of the old helix. It's important to keep in mind. We begin with one helix, it separates somehow, and the resulting daughter helices that are formed maintain the original halves of that parent helix, upon which, or opposite which, new halves are formed to create the new double helices. This hypothesis led to a specific prediction. The prediction was that if you could know which was old and new DNA after replication would occur, all the old DNA that was in the original parental double helix would now be dispersed between the two daughter helices equally. And the daughter helices would be composed of one half of old DNA and one half of new DNA. Now let's contrast that to the predictions we might have if we looked at these other two models. Think about the conservative model first. That model suggests that the DNA helix just remains intact. Once you've got it, you've got it. After replication, that model would suggest that the two daughter helices would be separately made up of, on the one hand, all old DNA, and on the other hand, all new DNA. In other words, the old DNA in the original parent is still in the original parent because the original parent is just copied intact, and the other daughter DNA helix is now completely new. The dispersive model made yet another prediction. That prediction was that the old DNA that was found in the original parental helix would just be randomly scattered, as its name suggests, across the two daughter helices. So we have three specific and different sets of predictions that could be used to distinguish between these three models of replication. The trick is figuring out how to know what's old and new DNA. Actually, it was several years, it was a number of years after Watson and Crick's original proposal before anybody could figure out how to experimentally test it. I mean, bear in mind that at that time, this was the hottest question on the planet, really. How does DNA replicate? Watson and Crick, through this remarkable model of the double helix that they had developed, had come up with a very neat hypothesis, a hypothesis that, if it were true, would provide a remarkably elegant molecular way to achieve this most important thing that a biological information molecule has to be able to do, and that is to accurately make more copies of itself. And the way to test this hypothesis was pretty obvious conceptually. Let's ask ourselves, after replication, what has happened to the material in the original parent's helix? We have very distinct predictions. The problem was figuring out how to know where old and new DNA was. This is often the way it is in science. An idea comes forward, people understand what they have to do, but the critical experiment isn't actually done until somebody comes along and says not, what's the experiment, but how can I actually do this experiment? And sometimes that can take years, as it did in this case. Eventually, researchers figured out an extremely clever way to know what the difference between old and new DNA is. And that's where we're going to begin next time with testing Watson and Crick's model.